Greetings, world, and welcome back to another episode of the NeuroWellness Project, a podcast devoted to exploring compassionate and evidence informed treatments for autism. This week, we are pulling back the curtain on one of the most fascinating yet overlooked frontiers of human health, the gut. The gut is far more than just a simple digestive organ. It is literally an entire ecosystem that communicates directly with the brain and nervous system. This gut-brain connection, which was once dismissed as fringe theory, is now being backed by a growing body of scientific research. And evidence is showing a clear link between the gut microbiome and autism, with some studies suggesting that up to 84% of individuals on the spectrum experience digestive dysfunction like leaky gut, dysbiosis, and other gastrointestinal conditions. And thanks to pioneers on the front lines like Dr. Sabine Hazan, it is becoming impossible to ignore the relationship between the microbiome and neurological conditions. Dr. Hazen is a world-renowned gastroenterologist, a highly accomplished scientific researcher, and a leader in clinical medicine with decades of experience exploring the depths of the human microbiome. Dr. Hazen is also the co-author of the book Let's Talk Shit, as well as the founder and CEO of both Ventura Clinical Trials, which is a research institute, and Progena Biome, which is a cutting-edge genetic sequencing lab that is examining the microbiome in the context of disease. And with well over 50 published papers in prestigious medical journals and over 300 clinical trials under her belt, Dr. Hazen is truly a force of nature. So how exactly does gut health affect autism, and what is there to do about it? Today, I have the honor of sitting down to talk shit with Dr. Sabine Hazan. Let's go. Dr. Hazen, thank you so much for joining us today.
Thank you. Thank you for having me.
So in the last clip, I just read off a summary of some of your accomplishments and qualifications, but I was wondering if you could maybe give us like a how you got into studying the microbiome.
It was really serendipitous, the whole thing. It was, I was the girl that was basically, I'm a gastroenterologist by trade. And I was the girl that was doing clinical trials for pharma. And I was the top recruiter for a condition called C. diff, Clostridium difficile, where basically I would recruit patients with C. diff. And then, if the clinical trial didn't work, I would do a procedure called fecal transplant or microbiota transplant. And what happened was when I was doing microbiota transplant, I was fixing C. diff and it was pretty successful. But then the reality is there were other things that I was fixing along the way. And when I started talking to all my colleagues and said, are you guys noticing other things? Like I had a case of Alzheimer's that improved with fecal transplant. And my colleagues basically were like, what? Yeah, I had alopecia areata. 2 guys grew hair. They were, bald and basically grew hair after fecal transplant. And then another guy said chronic urinary tract infections. Dr. Barodi in Australia was like having 2 cases of Parkinson's improving, but it wasn't consistent. Like he had Crohn's disease improving. It wasn't consistent and it wasn't reproducible. So what happened was with the case of Alzheimer's, I became obsessed, not because I improved Alzheimer's in that one patient, but mostly trying to understand why. Why did that happen? What microbe was the culprit of Alzheimer's? If there was a microbe, right? What was it in stools that changed this patient's Alzheimer's and he could remember his daughter's date of birth? So when that happened, I basically, started kind of like asking questions and go. I went to Dr. Sidney Feingold, who wrote the book on anaerobic infections in the gut, and again serendipitously he told me he was collecting stools of patients with Alzheimer's for 40 years, and he believed he found this microbe. He was obsessed with Alzheimer's, and so we kind of became friends. And he gave me this protocol, and he said, when you open your genetic sequencing lab, you're going to find this microbe and you're going to validate what I've been saying. And when he passed away during the Woolsey fire, he basically, the family called me and said, we're giving you all his books, his pictures, everything. And as I started rummaging through through the papers, um, and he was such a dear, like he had these amazing, frames that said, those who say cannot be done should not interrupt those doing it. I mean, he was just one of those like brilliant men. Anyway, so when I was looking through the papers and rummaging through the papers that he left me, there was a patent from him and Dr. Barodi on autism and fecal transplant. So both their names were on the patent. And I looked at that and I go, wow, Dr. Barodi. Dr. Barodi is the father of fecal transplant, right?
Mm-hmm.
And I said, autism and fecal transplant? Wow. And literally, so I took all the books. I wasn't going to open a lab, but I said, you know what? I was in the middle of a fire. I said, I'm going to put all the books in a room. I had like this whole, side of the building that was kind of empty. I said, I'm going to put it in a room I'm going to dedicate this room to Dr. Feingold. And I hung his frames and I put, in the middle of the Woolsey fire, I'm hanging his frames and I'm putting all the books. And so you could tell when my house is like, the roof is burning, but who cares? The backyard's burning, but who cares? I was obsessed about these books and these frames. My husband thought I was crazy. Anyways, so what happened was, I started the first day that I came back to work after the Woolsey fire. I had a producer call me and say, I have a kid with autism. It's the kid of the IT guy of our show. Can you help him? And I said, you know what? Why not? Let me help him. And I said, I can submit an IND to the FDA. I have a portal. I do clinical trials. Let me do that, right? And that was the beginning. That was the beginning of the saying, when you build it, they will come.
Yes.
People started reaching out to me. And I started talking to Dr. Barodi one-on-one. After I discovered that patent, I started talking to him daily.
Wow.
And basically said, you're going to be busy with autism. Wait till you see Dr. Adams' paper on autism. And I said, okay, why would I see autism? I only have one kid, right?
Mm-hmm, mm-hmm.
And sure enough, I started having a lot of kids in my office. And I said, and at that point I had already started the lab. And, it was kind of that moment in time where you go, should I spend my money on analyzing the microbiome in case I have Alzheimer's in the future?
Mm-hmm.
Or do I spend my money on a house in Italy, right? And I basically looked at that and said, you know what? I'm going to spend my money on understanding the microbiome because that could affect me later. That could affect my kids. And I never want to live a life where I regret things, where it's I coulda, shoulda, woulda, but I didn't because I was looking at money. Money is there to make us advance. And money is there, in fact, to help us advance science and medicine and understanding life. I'm in this, ever since I was little, my whole obsession, and I used to pray to God to say, where do we go after we die? And he showed me to the microbiome, which is essentially, microbes in your gut decompose your body and put you back into the ground. So, it's been kind of like, you really have, when you jump into the abyss of not knowing what you're doing at the beginning. I didn't know anything about genetic sequencing. I mean, that's not my world. I'm a gastroenterologist. I hired the scientist that was behind the BRCA gene. And basically that was behind like the research on the BRCA gene. And essentially it was one thing after another. Patients kept coming. We developed this assay. We started seeing things, seeing why I'm improving a patient with Crohn's disease by manipulating the microbiome. And then with the portal, we submitted that first protocol that second protocol for fecal transplant with autism. And COVID hit. And that's when my world changed forever because it became my protocol, my patent for COVID was the hydroxychloroquine, Z-Pak, vitamin C, D, and zinc. And
Wow.
when Trump talked about hydroxychloroquine, I looked at the sky and I just said, why me, God? Why me? Because I kind of like during COVID, I said, I have a genetic sequencing lab. I have a portal with the FDA. I do clinical trials for pharma. Who else can figure out how to survive COVID if not me, right? I've done like 300 clinical trials for pharma. I know a lot of things that a lot of doctors don't know. And so COVID was really, the courage. And I say this, it was faith over fear. It was, it was really trusting God that He's going to save you, and trusting God that He's going to show you the way out, right? And also being tested by God. Am I going to take the money for my patents, or am I going to continue my path? Am I going to take the money from my lab? Somebody offered me 185 million for Progenabiome, and I didn't take it. So because this is not about money for me, this is about being Indiana Jones and looking for that, the Holy Grail, right? And so essentially COVID was the beginning, and it was kind of the miracle. So that kid with autism took 3 years to get approved by the FDA because COVID was in the middle of it. But that case opened up the idea for me whether there's COVID in the stools, because if I was going to use his sister's microbiome, I wanted to make sure there was no COVID in there, because then I'm going to kill my patient, and then, I'm not going to find anything, right? Because we were in the middle of COVID. So the first thing we did during COVID was look for COVID in the stools. When we found COVID in the stools in 100% of patients that had positive nasal swab, it was a breakthrough. And then from there, we looked at the people that had severe COVID and the people that had, that were high risk, never got COVID. And we discovered that this bacteria called bifidobacteria. And then when you look at kids with autism, you discover that these kids are missing bifidobacteria. So to put it all together, because I know you're a mom with a kid with autism, that's how it was really serendipitous, the whole thing. It was really a curious person trying to find answers and being blocked along the way while finding these, but still moving. In other words, like, yes, keep coming after me, but I'm still moving. I'm not going to stop. I'm going to find another way. And, the pandemic, and I know you moms have gone through a lot with autism, because first of all, nobody, you weren't heard first, right?
Yes.
And
Yes.
second, there's not that much out there for you guys. You have to depend on, the functional medicine and the naturopaths, and you have to kind of see what other moms have done that works, and you communicate amongst each other, and you have a great motherly network, which I'm privileged to be part of, because, I feel for you. I feel for all of you ladies, because it shouldn't be that hard to move along a protocol. It shouldn't be that hard to have like 20 protocols on autism.
Yes.
1 in 12 boys in California have autism. We need to be waking up. My governor should be saying, we need to focus on autism. We have 1 in 12. This is not normal. Okay. Why is that not happening? So I knew during the pandemic that you ladies and fathers too, because there's a lot of fathers out there that are like, soldiers, warriors. But I knew that I was in good company, and that if I stepped in and started speaking and being a hurricane in research, I knew you guys would support me. And
Wow.
here we are.
Wow. Amen to all of that. Wow. Wow. Um,
Sorry, I tend to talk a lot, but there's a big story there. So it was kind of
no.
a loaded question.
Thank you. Thank you. So let's talk about dysbiosis in the gut. So it's my understanding that there's like three main types of dysbiosis. There's like an overgrowth of the harmful bacteria. There is a lack of beneficial bacteria, and then there's just an overall like loss of bacterial diversity in the gut. So, and you can have varying combinations of these, if I'm correct in that, but
So remember, let's go back a little bit. The microbiome space is at its infancy. There's no lab out there that has, that can say we are validated, reproducible, and we have clinical data. We're really writing the clinical data. Okay.
yeah.
Here's the thing about the microbiome. You cannot understand the microbiome until you reach a cure. You cannot reach a cure until you innovate and practice the art of medicine. We
Mm,
saw during COVID how many doctors stepped outside the box to treat COVID and innovated. Very, very few. So there's not many doctors out there that are trying different things outside the box to improve a condition. Okay. It's also very difficult out there because the problem is doctors have, there's a huge liability. You step out of that box, you're not protected anymore. You're going to get sued. You're going to
yeah.
have somebody report you. You're going to have somebody come after you. You're going to have someone, come to your office from the board of medicine or, or FDA or whatever, So
Mm-hmm.
anytime you step out of that box, it's very uncomfortable. So you got to really warrior up and lawyer up, frankly. So there's not that many doctors that are doing that. Okay.
Yeah. Yeah.
So there's nothing really out there that you could kind of hang your hat and say, this is what dysbiosis is. I can tell you there's a lot of hypothetical, right? I know there's a lot of naturopaths using a lot of different tests out there. But here's the problem with these tests. The majority of them are PCR. It's a piece of the microbes. The rest of them are very superficial sequencing. That means you're just looking at the surface, barely, what you ate last night, if that. So how does that help you when the microbiome is constantly changing? You're eating vegan one day, you're eating vegetarian one day, you're eating meat the other day. So you really need to kind of figure out the fine tune the assay or the stool testing to remove all these problems, right? So the majority of tests out there, especially if you're spending like $400, $500, I can tell you it's very superficial. You're not going to get anything out of that test. So what does that mean? Does that mean that that microbe is alive, or does that mean that microbe is dead? If the microbe is dead, what does it mean? You're evacuating it. That's what you're supposed to do. That's what your colon is supposed to do. So can you really hang your hat on a stool test that says you've got a lot of E. coli in there? Is that dysbiosis? Because you just, maybe the body's needing to, flush it down the drain, right? And therefore that E. coli is not really valid. People come to me all the time and they go, oh my God, doctor, my test shows I have H. pylori. And I go, do you have H. pylori? And then you end up doing an endoscopy on these folks, and you end up finding there's no H. pylori there. We biopsy it, we look for it. There's no H. pylori. So the test was flawed and told the patient. Now imagine the patient that believes in that test, which is not really clinically validated, and now goes to a doctor or a naturopath or functional medicine or internist or anybody, a GI doctor, and now gets treated for H. pylori with triple antibiotics, now just wiped out his gut. So now that patient was healthy, but
Mm-hmm.
because somebody told him he had H. pylori, he's now gotten the antibiotics to kill his microbiome, and now he's not healthy, right? So it's the same thing with the person that basically thinks that they have zero bifidobacteria. Bifidobacteria is that microbe that's in your probiotics, okay? So you look at your test and you're like, oh my God, I've got zero bifidobacteria. So now you're taking a lot of bifidobacteria. You probably had bifidobacteria, but now because you took so much bifidobacteria, you probably suppressed your ability to make bifidobacteria, to grow your own bifidobacteria. Kind
Wow.
of like what we do with enzymes, right? If I take enzymes, my pancreas doesn't need to work because it's saying, well, she's going to give me enzymes. If I take laxatives, my colon's not going to want to work because it's like, she's going to give me laxatives. Why do I need to contract and poop the poop out?
Yes.
I'll
Yes.
just wait for the laxative. So
Yes.
the body functions by us essentially letting the body do what it's supposed to do. So if I'm drinking a glass of orange juice, which is high acidic, I know my brain, my stomach's going to trigger a nerve impulse to the brain and say, hey, too much acidity. And then the brain's going to stimulate the stomach to say, send in the alkaline environment, right? So our body is, and then let's breathe heavier to kind of get that acidity out. Let's have our kidneys function to let that acidity out, right? So I think we need to, be more aware that the body needs to. So again, these tests are because they're not really validated. And Dr. Scott Jackson did a study at National Institute of Standards, by the way, where he showed that out of 7 tests, they were all showing different results. So how can you trust that if you can't have the same result? It's like you're going to the lab, you're bleeding, and you're getting your hemoglobin from one company and then another. But then guess what? You have different hemoglobins. Are you supposed to get that blood transfusion or not? Right? So so the technology of the microbiome is not there yet. I mean, I have an idea of what a gut dysbiosis is. I have to publish it first because that's how it works in medicine, right? You publish, then the next guy reproduces your data and says, you know what, Sabine is right. That's dysbiosis. But if I don't publish that, and the problem we have in medicine right now, and unfortunately, I am in a controversial topic, I'm treating autism, that they don't want to hear is a problem. And, I was the girl that basically showed loss of bifidobacteria and all these diseases. And I was the girl that showed the messenger RNA killed the bifidobacteria. So I'm controversial. So they don't want me out there. So because of that, there's a huge flux to retract my papers. So if my papers are retracted, how can I publish dysbiosis, right?
Yeah.
So we need to fix the retraction of the papers first so I can publish. And then once I publish, some other guy comes along and says, okay, Dr. Hazen, this is what dysbiosis is. Okay. Like the other thing about understanding the microbiome is really you need to do a lot of data, a lot of research. You need to understand what builds the microbiome, what kills the microbiome. You need to see what all these foods are doing to the microbiome to put it all together. Then you need to look at different races, different countries. This is not a small job. This is a big task.
Yes.
And this is a task that I embarked on, and I'm trying to bring in as many doctors as possible. But at the end of the day, it's not an easy task. And it really needs, I need my government to start working with me.
Yes, definitely. And let's touch on, you mentioned how most assays out there are just kind of surface level, like superficial, right? This is where Progenabiome comes in. So the genetic sequencing. Could you
Correct.
maybe give us a little blurb on how that differs from your run-of-the-mill general assay that you would get?
So without telling our secret sauce, because we've done a lot of work and, it's important to have a validated assay. We go super deep into the microbiome and we have some clinical data. We don't have all the clinical data, but we have some clinical data. We have a huge database. Um, and, and here's what it takes. It takes taking a good history of the patient. Because when you look at these samples and if you see that there's something that doesn't match with the other guy, you got to go back and call the patient and say, are you sure you didn't take an antibiotic before? Are you sure you didn't take probiotics? Because it doesn't make sense. I have like 20 samples that look like that, and you're kind of like that off the beaten path sample, right? That's what it takes. It takes diligence. It also takes a physician, not a robot, to basically develop that relationship with the patient to know what they're taking. Because a patient that takes marijuana or drinks alcohol and doesn't tell his doctor is not the same as a person. So a person who has Alzheimer's and drinks alcohol, and a person that has Alzheimer's and doesn't drink alcohol, they are not the same. One is wiping his gut with alcohol, the other one has something else. So you can't really compare the two. So you really, really need a lot of samples, and you really, really need that meticulous protocol that basically says, you know, what's an apple, what's an orange, so that you
Yeah.
can compare oranges with oranges and apples with apples. Otherwise, listen, in the microbiome, it's a huge rabbit hole. And I see people all day long falling for the rabbit hole, people that come out with, oh, Crohn's disease is this microbe. And I look at that and I go, are you sure it's this microbe, or did you contaminate? I think you contaminated. So, there's so many studies out there that I look at every day. And then I look back at my database. I'm like, nope, that's not the right microbe. Sorry. So the problem with that is that if I don't see that microbe, and you're telling me that the disease is that microbe, here's the problem. You're going to go down this rabbit hole, and all the scientists are going to follow you. And then you're never going to find an answer for MS because you're down the rabbit hole. So it's important to work together. As much as everybody wants their own lab, everybody wants to have their own thing, I'm going to tell everybody who's doing anything, there's enough business for everybody in the microbiome space. We're looking at trillions of microbes, and
Yeah.
we've barely touched the surface on what microbe is good and what microbe is bad. And when we talk about autism, there's a whole gray matter of microbes there that we don't even know their names. Dr. Feingold
Mhm.
named a bunch of microbes. Elistopes feingoldii was named by him. So there are a lot of microbes that we need to start discovering that are out there that we've not even discovered. So I think it's important to work together to see what we all have, and we need to collaborate together for this big, massive project. And it's not a Sabine Hazan or Progenabiome project. It really needs to be a world project where Japan collaborates, China collaborates, so that we can all see, what does the microbiome of a Japanese look like compared to a Japanese in America, a
Yes.
Japanese that lives in Japan versus a Japanese that lives in America. What microbes have they lost when they came to this country?
Yeah.
So this is very important, and we need to have the same standardized operating procedures so we can have the same findings. Otherwise, we're lost. So that's my project. It was initially thought of being, it was supposed to be done by a company, if you recall, Ubiome,
Mhm.
but unfortunately, it didn't work out because they were doing some stuff that was not so clean. But here's the thing: all those scientists that were at Ubiome, I encouraged them to join Progenabiome. It doesn't have to be the end. We can continue together.
Yes.
I'm
Yeah.
not a destroyer. I'm a builder, and I think we need to, instead of killing the data, prove me wrong, and let's work together. Right? That's because guess who's hurting when we don't work together? Your kids, our kids, the future generation that's going to take care of me when I'm getting older, and is that generation going to be around when I'm getting older, or am I going to have a robot taking care of me? So
Mm.
we need to preserve humanity. We need to preserve the microbiome of humanity, and we need to unite as humans, because the microbiome of humanity is disappearing, sadly. And with it, humanity. I see it already. The two most important microbes,
Mhm.
um, we out of like 1,000 samples, one of them had, an important microbe that helps absorb calcium. And then less than 5% had an important microbe that helps absorb sugar. So, what happens when those microbes disappear and we've killed off every single one of those microbes, and probiotics are not the answer because they're not in grafting, or
Yes.
we just can't make enough probiotics? That's a problem.
That's a big problem, definitely. So, your work is definitely kind of like challenging that traditional view about autism, right? That it's just purely genetic, it's purely neurological. So, could you tell us a little bit about how perhaps like
No, I just think like autism is probably multifactorial. Yeah, everything in medicine has a differential diagnosis. You know, your
yes,
person comes in vomiting, it's not necessarily just an ulcer. You know, it
yes,
could be an obstruction, you know, it could be cancer, you know. So you got to always have in your mind a differential. So the differential with autism could be that it's a brain primary in some kids. It's a connection problem from brain to gut primary. It's a gut problem, gut dysbiosis, or it's a genetic problem, mutation, perhaps, may even be a coagulopathy problem. It may be a, you know, so we need to be better at opening the narrative. And here's the thing. If something is not fixing a kid, so look at Dr. Adams' data, for example, 50% improve with autism, right? Why didn't the other 50% improve? Do
yes,
those kids have a neurological? Do those kids have a genetic problem? We need to understand, we need to go to the failures to understand the disease better. You know,
yes,
I think when we stop and we say we have a cure for autism, you know, and here's a pill full of poop, and that's the answer. No, it's not the answer, you know?
yes, absolutely. I don't know if this is kind of like a loaded question, kind of like touching on that note, but from your perspective, these like microbial variations that are in, everybody, do you think, especially with autism, that it's like a cause that's contributing to the development of autism? Do you think it's more likely just like a correlation?
I think it's a risk factor. I think it's a risk factor. I think what we're going to come to find out in autism is that there are multiple risk factors. I think it's not just a one thing. I think it's an accumulation of things. The think about life now. Mom is super, you know, I was telling, you know, my daughter was saying to me, she goes, you know, it's really unfair for women. We have to look good, we have to exercise, we're taking care of our kids, we're bringing money, we're working, you know, like as she sees me like I'm juggling 25 things at the same time.
Yeah.
And she's like, it's not fair for women to have this whole big thing, you know, all this responsibility, all these responsibilities, right?
Mhm.
Um, when you think about this, the pressure that we put on ourselves as women, right? The job, the working, and being pregnant. In the olden days, you would stay at home, and a lot of women are doing that, and I commend them for that. You know, we wanted, and I commend the woman that wants to work as well, right? Because at the end of the day, freedom of choice. You want to be a doctor, you want to be a lawyer, you want to be an astronaut. Good
Yeah.
for you. You're a human. You should have a right to do whatever you want to do. And you
Abs-
want to have kids, if you can juggle it, go for it. You know what I mean? That's
Yeah.
the beauty of women, is that we can multitask and we can do it all, in my opinion, and we do it all in heels. Um, for some of us, anyways. So the key is mom is stressed. She has a job. She's pregnant. Now, that already kind of kills your microbiome, the delivery. You're always on the go. You're eating that sandwich really fast. You're not calming down the system. Then mom is delivering the baby, C-section, vaginal. We've seen that C-section is a different microbiome than vaginal. Then the baby gets that antibiotic cream in his eyes. First thing, right? Because we want to avoid an infection in the eyes, right? What is all that doing to the microbiome? Then some babies are getting antibiotics because
Hmm.
mom got C-section or
Yes.
episiotomy, and they
Yes.
get pushed antibiotics. So some babies end up in the intensive care unit and they get antibiotics and
Yeah.
they're on oxygen and everything. What is all that doing to the microbiome? And then you've got the, you know, vaccine question, right? Is
Yeah.
the vaccine a culprit or not, right? And I think the vaccines came on as a risk factor because moms noticed, especially after the MMR, that their kids were developing properly, but then after the MMR, smack, the kid is like looking at the fan, or the kid stops developing. So they correlated the MMR to that autism. So to moms who are not all, doctors and scientists, they're like, the MMR was the culprit, right? That doesn't mean it was the MMR. It could be just a prolongation. In other words, like what got you up to here. In other words, you killed a little bit with work, you killed a little bit with the antibiotics, you killed a little, and then bam, MMR hits, and you're down to your kid has zero bifidobacteria, gut is desolate. Now he's exposed to grandma who has a cold. He's exposed to dad who's, got whatever microbes. He's exposed to mom who's anxious. By the way, we wrote, we published on anxiety, which is the difference in microbiomes between anxious versus non-anxious. And there's actually a group of microbes that's really elevated in anxious moms and or anxious women or men, whereby you have to ask yourself, is the Bacteroides, because that's the microbe, is the Bacteroides a or increasing because of the anxiety that the person is under, or is it the Bacteroides creating the anxiety, right? Regardless, we noticed like a lot of Bacteroides with anxiety. And, so I always, when I see that and I see my babies have a lot of Bacteroides, I know they're getting it from somewhere. So, they've lost those good microbes and now they're exposed to mom who's super anxious about the baby not developing, and then is giving her microbes to the baby. And so you almost have to kind of treat the whole family in a way with autism, because you have to restore that balance. You also, and here's the other thing that nobody really talks about, or a lot of people talk about, but not mainstream media. Is there mold in the house? Is there toxins? Do you have a plant that's secreting? Do you have a cat that's secreting some bacteria that your kid is getting, you exposed to? Do you have a dog that's the same thing, So because animals share our microbes, right? We all are a vacuum. So, the whole thing is very interesting. And I think we're going to come to find out there are risk factors. I think every one of those steps needs to be analyzed, in my opinion. The moms, like if I had multiple amount of money, I would be doing a study where I look at all the moms, the stools of the moms and the stools of the babies, and I would look at C-section versus vaginal. And then I would look at whether they took those creams of antibiotics in their eyes or not. I would look at antibiotics or not, and I would look at vaccines before and after to see what is happening. You have a baseline of a good microbiome, and then you give the vaccine, you kill off the bifidobacteria and the microbiome. Is that what we saw with COVID anyways? The messenger RNA kills the bifidobacteria. So is that why people are getting COVID, a second time or a third time right after the vaccine? Is that why people are getting neurological problems, herpes encephalitis, CMV encephalitis after the vaccines, coagulopathy? There's going to be a time where we're going to start thinking about heart disease as a microbiome problem, as a microbe. And I think it's time to start looking at that. So I'm excited for the future because I'm kind of, I'm leading this path of just talking shit. That's my book, Let's Talk Shit. But I think it's waking up doctors. I really think it's like they're paying attention. And that's what we need. We need doctors to pay attention. Prove me wrong. That's it. Prove me wrong that there's no microbe that's linked with heart disease. Prove me wrong that there's no microbes linked with autism or Alzheimer's or Parkinson's or cancer. I think this is what we need to be better as humans, because the world is suffering. And we're having also, I mean, we've talked about all these diseases, but what about mental health? Mental health is through the roof. People are depressed, they're anxious. What are we doing? What are we doing to humanity? It's sad.
It is. Yeah, it is. But, you're out there, you're doing the good work. And thank you so much for that. Are we allowed to talk about what reflorization is? Can I ask
So
you?
refloralization, yeah. So refloralization is really a term that's bringing back the flora. So whether bringing back the flora from smelling that essential oil, or bringing back the flora from being in your garden and touching the dirt, or bringing back the flora from drinking that good glass of milk, or that kefir, or that yogurt, or eating good food, and it's bringing back the flora. It's stopping to destroy and refloralize. So I never liked the term fecal transplant. And I also
Mhm.
don't like to play with stools. Let's be honest. I
Mhm.
don't like taking stools from a healthy donor, mixing it and then implanting it. It's disgusting. It smells. Nobody likes to do that. So if I can figure out a better way to refloralize the gut, to bring back that good flora in the gut, then it might be a better solution than taking stools from a healthy donor and putting it into an unhealthy. Here's the other problem is we're running low on good donors. We have a huge population that is, I can tell you, probably out of like a couple thousand samples, there might be two that have a good enough microbiome to use as donors.
Yeah.
So when people come to me and they're like, "Oh, I'm perfectly fine. I don't take antibiotics. I've not gotten COVID. I'm the perfect donor. I'm gonna be your golden ticket." And I test their stools and I go, "I'm sorry, you're not." So it's like the Willy Wonka and the gold ticket. It's very difficult to find that golden ticket. So, and those who have it have worked really hard at it. And those people are resilient.
Absolutely. So I've seen other presentations that you have given, and you touched on it in the beginning, where you say that when parents are trying to like put a band-aid on a leaky gut, right, in their child with autism or what have you, and they're giving their kid all kinds of vitamins and supplements and probiotics and detox, there's so much out there, even like restrictive diets, that maybe that's
Right, right.
not doing them, maybe that might be doing them a disservice.
I,
So
I think the big mistakes that a lot of parents are doing is they're jumping ship. They're going from one to another to another. They see nothing's help. They see like, "Oh, well, I gave my kid this probiotic, but it's not helping. Let me go to
yes,
another doctor and try another protocol.
yes,
Oh, I did this protocol, but it didn't work. So let me go." But the problem is with the microbiome is it takes months to years to fix. So you're not necessarily going to see the changes. You can only see the changes at the microbiome level, where you basically can say, "Oh, look at that. My patient improved their microbiome." I can tell you, even me, when I
mhm,
do fecal transplant, I've done it on one case of autism,
mhm.
and we used the sister's microbiome, and it engraft at the beginning, so it did show me engraftment. But for changes to be seen, it took 6 months to start seeing the kids start improving. And here's the thing, at some point, you need a lot, you need more. It's not a one-time deal.
Yes, absolutely.
But you have to convince the FDA to keep doing it. And the problem is that there's no data on that. So who's going to do it, to show the data that you need
Exactly.
reoccurring, FMT?
Exactly. So what advice might you give to a parent who is attempting to navigate their child's autism journey through gut health, through the lens of gut health? What would be your best
Well,
piece of
the
advice?
first thing,
Mhm.
yeah, the first thing I would say, and the simplest, is get yourself some good vitamins, good food, good nutrition. Okay. Make sure and remove all the junk, and make sure you know your farmer, make sure you know where you're getting your milk. That's the first thing. Decrease the stress in the household. Keep your kid as best as possible. Um, and then, embark on one of the clinical trials that are out there. We certainly have clinical trials on autism where we can test the microbiome. Those who can afford to test, usually, join us in the clinical in my clinic, and those who cannot afford to test, we put them on a wait list till our foundation raises money to test their kids. So that's how we kind of, people volunteer to pay for testing for their kids, understanding that it's research and understanding that this is in a way their contribution to the research to see what their kid looks like, and then for us to use all that data to help the kids.
Absolutely. And if somebody wanted to find more information about your book, about Progenabiome, where's the best place that they could go to find out more information about you?
So progenabiome.com, everything's in there, publications are in there. I encourage people to support the microbiome research foundation.org. That's how we're able to submit INDs to the FDA. The first IND cost roughly $300,000 for one kid. The second IND, which is for 30 kids, we're up to $400,000 already spent, and we've yet to be approved. So it is extremely expensive. The FDA lawyers are $1,500 an hour. The regulatory personnel to submit those paperwork are $400 an hour. This is not an easy process. The stool testing is expensive. The writing the protocol is expensive. The writing the consent is expensive. The, electronic consents we have are expensive. So everything in this is expensive. So please donate to the microbiome research foundation.org.
Dr. Hazen, thank you so much. This has
Thank you.
been
Thank you.
so fascinating and just what an honor. Thank you so much. And thank you to everyone who watched this episode. We will catch you in the next one.